Pharmaceutical companies spend, literally billions of dollars, right?
Right.
Just engineering drugs to hit one very specific biological target perfectly.
Oh, absolutely. It's the holy grail of precision medicine. We're taught to view these targeted therapies like sniper rifles.
Right. But what if, and hear me out here, what if the secret to treating our most stubborn, resistant cancers is actually relying on drugs that are beautifully, surprisingly sloppy?
Yeah, it fundamentally upends how we look at pharmacology. We've spent the last 2 decades trying to engineer the messiness out of our cancer drugs.
Exactly.
But the research we're looking at today suggests that very messiness isn't a flaw in the system. It is actually a feature we can exploit.
Right. So let's set the stage. We are pulling from a really groundbreaking April 2026 paper published in Nature Biotechnology.
And it is a massive study.
It really is. And our mission for this deep dive with you today is to explore how polypharmacology, which is the phenomenon where a drug designed for one specific target accidentally hits several others, is unlocking treatments for diseases we previously thought were untouchable.
Yeah, to understand the sheer scale of what this research team accomplished, we kind of have to look at the genome, just visualize this massive glowing map of cellular pathways.
A map of switches, basically.
Exactly. Protein kinases are the communication switches in our cells. They regulate everything from cell division to apoptosis, which is programmed cell death.
So when these mutated kinases get stuck in the on position, you get runaway cellular replication.
Right. You get cancer.
Okay, let's unpack this. We usually think of drugs as a lock and key, right? Specific drug key for a specific kinase lock. What happens when a key accidentally opens 20 different doors?
Well, historically, drug developers ran away from that. There are over 500 kinases in the human genome, and their ATP binding pockets, the actual locks, they look structurally very similar.
So a key for one door is almost guaranteed to rattle the locks on a few others.
Precisely. And for years, scientists relied on computational binding simulations. If a drug looked like it hit multiple targets on the computer, they assumed it would just cause lethal off-target toxicities in a human patient.
Wait, isn't off-target usually just like a polite word in pharmacology for a terrible side effect? Why are we suddenly celebrating this?
What's fascinating here is that an off-target effect is only a toxicity if it hits a crucial healthy pathway.
Oh, I see.
Right. If it hits a pathway that happens to be driving a completely different type of tumor, it's not a side effect. It's a drug-repurposing goldmine.
So we can basically skip the decades of safety testing and use existing drugs for entirely new diseases.
Exactly. So the researchers stopped guessing with computer models. They took 92 clinical kinase inhibitors, and 86 of these are already FDA-approved, sitting on pharmacy shelves today, and physically profiled them against 758 different kinases.
That's 409 normal wild-type kinases and 349 mutated cancer-driving variants.
Right. But the methodology here is the real game changer. They didn't just look to see if the drug physically stuck to the protein. They used a functional hotspot assay.
Which is so crucial. A kinase's entire job is to grab a phosphate group from an ATP molecule and attach it to another protein, right? Like passing a baton.
Yes, passing a signal. So the researchers used radioactive ATP. They put the isolated kinase, the drug, and this radioactive ATP in a well together.
Oh, wow. So they're tracking the actual radiation.
Exactly. If the kinase works, it transfers the radioactive phosphate, and the target protein becomes hot. If the drug successfully blocks the kinase, the transfer fails.
They are measuring the physical failure of the biological switch.
Yes, and they ran this at concentration of exactly 1 micromolar.
Which I should add is really important because that mimics the actual physiological concentration in human blood plasma after you swallow a standard pill. You're talking about real-world conditions here.
Real-world conditions. And the data from that assay fundamentally rewrote the map. Before this paper, those 92 drugs were documented to hit 89 primary target kinases.
Okay.
After running the physical hotspot assay. The landscape of druggable targets exploded to 235 kinases.
That is wild. Okay, so to recap really quickly, bullet 1, kinases are cellular switches that cause cancer when stuck on. Bullet 2, the hotspot assay used real radiation to prove drugs hit way more targets than computers predicted. And bullet 3, our known druggable targets just jumped from 89 to 235.
A massive expansion of our arsenal.
So here's a question for you listening. If you had a master key that opened unintended doors, would you throw it away because it's messy, or would you meticulously map out every single door it opens?
I mean, any rational scientist starts drawing a map.
Right. And when you look at the map they generated, you immediately start seeing solutions to diseases that have historically been absolute dead ends, like glioblastoma.
Yeah, glioblastoma is a highly aggressive brain cancer. It's notorious for evading treatment and has devastatingly low survival rates.
And in a subset of these tumors, the driving force isn't your typical growth factor. It's a pair of kinases called IRAK1 and IRAK4, right?
Exactly. Now, normally, these IRAK kinases are integral to your innate immune system. They mediate inflammation. But when they are highly expressed in a brain tumor, they drive cancer progression.
And let me guess, there are currently 0 FDA-approved drugs designed specifically for IR1 or 4.
None. But when the researchers cross-referenced these kinases against their new polypharmacology map, an off-the-shelf drug popped up, tepotinib.
Tepotinib, wait, isn't that an established drug for lung cancer?
It is. It's an inhibitor for the MET kinase, primarily for non-small cell lung cancer. But the assay proved it is also a highly potent inhibitor of both IRAK1 and IRAK4.
And the cellular mechanics of this are just fascinating because they didn't just stop at the enzyme level. They took tepotinib and applied it to live glioblastoma neurospheres.
Right, these 3D clusters of actual human tumor cells. They even tested it in live mouse models. And the lung cancer drug didn't just passively halt the brain tumor's division, it literally altered the tumor's cholesterol metabolism.
Yeah, a tumor cell dividing that rapidly needs immense amounts of cholesterol, right?
Yeah.
To build new cell membranes and lipid rafts for signaling. It's ravenous for fat.
Exactly. And tepotinib basically forces the tumor to empty its own pantry. The sequencing showed the drug upregulated specific genes ABCA1 and ABCG1.
And those are efflux pumps, right? They physically pump cholesterol out of the cell.
Yes. And at the exact same time, the drug suppresses pathways regulated by NF-kappa B and SREBF2.
Which normally tell the cell to manufacture more cholesterol.
Right. So it turns on the exhaust pumps while simultaneously shutting down the manufacturing plant. They could actually measure the cholesterol levels rising in the fluid around the cells as the tumor pumped out its own building blocks.
It's like realizing the plumbing wrench you have is actually perfect for cutting the electrical power to a highly secure vault. You're starving the tumor of the materials it needs to build itself.
It really highlights how artificial our neat little biological categories can be. An inflammation switch governs the lipid diet of a brain tumor.
Incredible. So, quick recap here. First, glioblastoma is sometimes driven by IRAK inflammation switches. Second, tepotinib, a lung cancer drug, accidentally blocks those switches perfectly. And third, this blockage literally starves the brain tumor of cholesterol.
It's a brilliant proof of concept.
Absolutely. So think about how connected our biological systems are. Does it surprise you that an inflammation switch controls a tumor's cholesterol diet?
It shows why targeting a single pathway often fails in the clinic, because tumors are incredibly adaptable. If you cut power to the main system, they usually have a backup generator waiting to kick in.
Ah, the paralog problem. Biology loves redundancy.
It relies on it. Paralogs are basically redundant backup kinases. They arose from gene duplication during evolution. They're twin switches.
So if you block kinase A, its twin kinase B just takes over the workload.
Exactly. Single gene knockout studies often miss these because of this functional overlap. The target is inhibited perfectly, but the tumor doesn't care.
Which is where a sloppy drug becomes a massive advantage. Let's look at the case study they did on pancreatic cancer.
Right. There are two paralog kinases here called MRK2 and MRK3. They drive the YA-hippo signaling pathway.
And YAP is a powerful signal that tells the cell to survive and multiply, right?
Yes. In pancreatic cancer, if you silence just MRK2, MRK3 keeps the YAP signal flowing. You have to take out both simultaneously.
Which is super hard to do with precision engineering.
But the map revealed that brigatinib, a drug approved for ALK-positive lung cancer, powerfully inhibits both MRK2 and MRK3 at the exact same time. It shrank the pancreatic tumors in mice by taking out both the primary system and the backup generator.
Here's where it gets really interesting. They also used something called the DEP map to find more of these double-agent drugs, didn't they?
They did. The DEP map, or dependency map, is this massive database where scientists use CRISPR to literally knock out individual genes across hundreds of cancer cell lines.
Right. So if deleting a kinase causes the cancer to die, you know that cancer is dependent on it.
Exactly. So they cross-referenced this DEP map with their new drug data, and they looked at neuroblastoma, a really devastating pediatric cancer.
Driven by a protein called NMYC.
Right. And NMYC is notoriously slippery, It lacks deep binding pockets, so you can't drug it directly. Plus, it degrades very quickly on its own.
So how does the tumor keep it around?
It relies on aurora kinases, specifically aurora A. The kinase physically binds to NMIC, acting like a scaffolding that prevents the cell's waste disposal system from degrading it.
Oh, wow. So if you destroy the scaffold, the deadly NMIC protein falls apart.
Precisely. And they found that axitinib, which is a blood vessel drug used for kidney cancer, actually hits these aurora kinases potently.
So they applied the kidney cancer drug to the pediatric neuroblastoma cells, and it just kicked the scaffolding away. The NMIC degraded, and the tumor cells died.
It's beautifully sloppy.
Okay, time for another quick summary. Bullet 1. Tumors use backup twins called paralogs to survive targeted drugs. Bullet 2. Brigatinib hits both MRR twins in pancreatic cancer. And bullet 3, cross-referencing with the DEP map showed axitinib destroys the survival scaffolding in neuroblastoma. Spot on. So if you were designing a cancer therapy, would you prefer a highly targeted sniper rifle or a net that captures both the target and its inevitable escape route?
Given the plasticity of cancer, you definitely want the net. Because tumors don't just rely on backup generators, they can undergo radical physical transformations to evade drugs entirely.
You're talking about EMT, epithelial mesenchymal transition.
Yes. It's the mechanism behind metastasis and severe drug resistance.
So epithelial cells are normally stationary, right? Like tightly packed bricks in a wall.
Exactly. But during EMT, a tumor cell alters its gene expression. It loses that rigid structure and acquires the migratory, squishy properties of a mesenchymal cell.
It essentially shapeshifts to become a mobile stealth unit. And once it's in that state, standard chemotherapies just bounce right off it.
The researchers wanted to see if any of their MAP drugs could target the specific kinase networks that maintain this mesenchymal state. They looked at a highly aggressive breast cancer cell line called MDA-MB231.
And the map pointed them to gilteritinib, which is normally a leukemia drug.
Right. But it broadly inhibits these mesenchymal kinases. When they treated the breast cancer cells with it, it forced the cells to literally revert from the tough mesenchymal state back into a fragile epithelial state.
Wait, it reversed the shape-shifting.
It reversed it. They saw a massive upregulation of E-cadherin, which is the molecular glue that locks epithelial cells together, and a downregulation of vimentin, which gives mesenchymal cells their mobility.
So it stripped off the armor, and that made the cancer sensitive to another drug, cobumetinib, all over again.
Exactly. It proves that acquired drug resistance isn't always permanent. But, they also looked at the most direct form of resistance, mutational rescue.
Right. This is when the tumor mutates the actual lock, the amino acid sequence. So the specific drug key literally cannot fit anymore.
Yes. They tested 349 different mutations and fusions. And the data showed that a staggering 94% of mutations and 97% of fusions can be inhibited by at least one existing drug on their map.
94%. So almost every mutation already has a key that fits.
Yes. Take the MET kinase. There's a mutation called D1228 that resists all normal Me2 drugs. They just hit a physical wall and can't bind.
But gilteritinib, that same leukemia drug, has the flexibility to slide right into that mutated pocket and kill it.
Exactly. And resistant FGFR mutations can be hit by erdafitinib or selpercatinib, which are bladder and thyroid cancer drugs.
If we connect this to the bigger picture, drug resistance isn't always the end of the road. It just means we need to look at a different part of the map.
It changes the entire paradigm. The bottleneck isn't always inventing new molecules. It's finding the ones we already have.
Okay, final mini recap. 1, EMT is when tumors shapeshift to become resistant, but drugs like gilteritinib can force them to change back. 2, a massive 94% of cancer mutations already have an existing drug that can block them. 3, existing drugs from entirely different cancers can rescue patients with resistant mutations.
Perfect summary.
So how does knowing that 94% of cancer mutations already have an existing molecular key change the way you think about the future of cancer treatment? It's huge.
It's massive. And the best part is the researchers didn't just publish this and walk away. They created the KiRHub.
The Kinase Inhibitor Repurposing Hub, a completely public web tool.
Right. It democratizes the data. Anyone can log in, input a specific genetic mutation, and instantly map it to existing FDA-approved drugs.
So what does this all mean? We started out talking about precision, the sniper rifle. But this deep dive really shows that polypharmacology, the inherent messiness of biology, isn't a flaw.
No, it's a feature we can exploit. We spend billions inventing new drugs from scratch. But looking at this map, you have to wonder, How many cures for rare or resistant diseases are already sitting on our pharmacy shelves waiting for us to realize what they can really do?
That is such a profound thought. The answers might already be in our hands. We just needed a better map. Thank you for joining us on this deep dive, and to everyone listening, keep questioning the obvious.